Hello, it's great to be with you today, albeit virtually. The Biochemical Society and Portland Press are pleased to welcome you to this webinar, which is part of our Biochemistry Focus webinar series. Topics in the series will include different research areas in the molecular biosciences, as well as practical sessions to support career development. Each webinar will give you the opportunity to ask questions via text, and we welcome suggestions for future topics and speakers to feature in our webinar series. Please see the website for more details. So my name's Anne Osborne. I'm from the John Innes Centre in Norwich, and I'm a plant biologist. I work on natural products. Today's webinar is called Developments in Industrial Biotechnology. The application of biotechnology to industrial processes is transforming the way that we manufacture products. By maximising and optimising existing biochemical pathways, we're able to integrate the improvement of products with pollution prevention and resource conservation. I'm delighted to be joined today by 3 researchers working in this area with the aim to deepen our understanding of the potential of industrial biotechnology and ultimately create the technologies required to implement a more sustainable bioeconomy. So first we'll hear from Dr. Leonardo Rios Solis, a lecturer in synthetic biology and biochemical engineering at the Synthesis Centre for Synthetic and Systems Biology at the University of Edinburgh, where his lab's research motivation focuses in promoting further understanding of the interface of biochemical engineering and synthetic biology, as well as to find novel ways to apply this technology to tackle the growing socio-economic inequalities that plague the world. Today, Leo will present his group's work on the use of microscale bioreactors to enable the high throughput testing required to characterise novel constructs and engineered strains in gene synthesis and DNA assembly. So, hi Leo. Our second speaker will be Dr. Katalin Kovacs, a senior research scientist at the Synthetic Biology Research Centre, Nottingham. This centre concentrates on engineering bacteria to make industrially useful products from C1 feedstocks, including the greenhouse gases carbon monoxide, carbon dioxide and methane, with the overarching ambition to reduce societal reliance on petrochemical-based feedstocks by creating the technologies required to implement a more sustainable bioeconomy. The centre is developing genetic tools that will allow for the engineering of bacteria capable of producing chemicals that are currently only available from fossil fuels. Katalin will today talk about the metabolic engineering of the chemolitho-autotrophic bacteria Cupriovidus neckata H16, the centre's chosen chassis for research into aerobic gas fermentation for the sustainable production of monomers and biopolymers. So hi Katalin. And then finally, we'll welcome our third speaker, Dr. Mark Dunstan. Mark's a Senior Experimental Officer at the Synthetic Biology Research Centre for Synthetic Biology of Fine and Specialty Chemicals, SynBioChem, bringing together interdisciplinary expertise from across the University of Manchester. SynBioChem is applying synthetic biology approaches to engineer microbes for rapid, predictable fine and specialty chemicals production. Their 3 main challenge-led projects aim to accelerate the production and scale-up of diverse and novel alkaloids, flavonoids and terpenoids, which represent key chemical scaffolds with diverse commercial interest. And Mark's talk today will outline the recent development by SynBioChem of a pipeline for the rapid prototyping of microbial production strains towards the production of material monomers and specialty chemicals, focusing on engineering E. coli towards de novo production of gatekeeper 2S flavanones. So before I hand you over to our speakers, please note that the questions will be asked at the end of the webinar, but you can send in your questions anytime during the talks. And if you have a question, please type it into the question box as shown in the image on the screen. It would be helpful if you could also indicate which speaker you're directing your question at. So with that, I'd like to hand over to Lee and just check that his slides are okay. Are you able to share?
Perfect. Thank you very much for that nice introduction. And yes, so my talk, then yeah, I will focus in microscale tools, as in the title, to produce microbial cell factories, which is what we do in my lab. We try to create those microbial cell factories to produce high value products, but we're really trying to make it in a way that they can work at industrial scale conditions, and that we can then also engineer them in a way that we can understand what is happening, and that's not an easy task to do at all. And then that's why it has to be run in a really multidisciplinary approach. Industrial biotechnology, more than I think anything else, it really needs to involve, and that's why I'm lucky to be working at the CINCIS, the Center of Biology, where part of what we're talking about now, it involves people working from in automation, informatics, engineering, chemistry, but also social science and entrepreneurship to really have this important impact in industrial biotechnology, medicine, healthcare, for example. So why do we have a nice ecosystem here in CINCIS that it's allowing us to try to engineer, and we're following this well-known synthetic biology iterative approach. And here, for example, we're quite proud of our Edinburgh Genome Foundry, where, for designing the DNA, assembling it, multiplexing. So we have done lots of advances in that. And part of my lab, we're still working a lot in that, and new ways to engineer the cells. But what we have noticed is the next step, especially cell phenotyping, is where we have some problems there, or some bottlenecks, especially for industrial biotechnology. And it's not that we cannot really characterize the cells properly. I mean, we have, for example, genomics in for metabolite metabolomics, for example. We can really measure all the metabolites inside the cells. Or Edinburgh Genomics for transcriptomics, proteomics, and genomics. We can sequence all those cells fantastically, and not so expensive in many cases, or even sometimes in real time. But the problem is we're still doing it at the wrong conditions. We're still doing it at lab conditions that don't mimic industrial scale conditions. We're still doing it with the feedstocks that doesn't really mimic the industrial scale conditions. So sometimes all that effort, all that data that we get, and we analyze to do this cycle, we optimize in different conditions, and then it's not been really optimal, which is a little bit something that I'm gonna talk about it. So, what are we proposing here in my lab? How are we trying to tackle this challenge? Is okay, we're designing the cells. We're using robots, plasmids, bacterial DNA, as I said, with the Genome Foundry. That's a totally different area I'm gonna focus now. But then, okay, the problem is here when we're testing those cells. A really nice, fantastic tool are the microplates. So we can really have this high throughput screening. But as I said, microplates, they don't really predict the shear stresses we get at the industrial conditions. We don't predict many of those. It would be quite nice to be able to do some mini more iteration cycles in here, or find methodologies that we could really, since the first iterations, predict those behaviors under the industrial scale conditions. Some new advancements that we use in my lab, for example, are the probolector, or plates with special shapes, like flower shapes, so you can get more oxygen. And now, instead of like before, we were always testing our cells with the plate reader, for example, and you would only get ODs of 1, 2, 3. That doesn't predict at all industrial scales, So at least here we can get ODs 40, 50, 60, 70. It's more similar to an industrial scale bioreactor, and believe me, just by different higher ODs, things totally change. Different stresses, the wastes are different, communication between cells that we don't even understand are happening. So we can start to include those conditions here. Other, for example, we can start adding fed batch methodology with that type of microplates, which is what we're always using in the industrial scale. But fed batch, it totally changed some of the what's happening inside the cells. So that's something. And things that we cannot yet do is maybe controlling, for example, the oxygen concentration in a small scale. Sometimes pH too, shear stress is difficult, but that's some of the things that we're working to incorporate in our lab. And the key thing is then, yeah, all that data that we're gathering is telling us of how the cells respond to some of those stresses and industrial escape conditions. And then, yeah, we can understand, we can target it, we can learn how to then solve those problems. And this is also when many times there are things that we still don't understand, and that's where artificial intelligence at least is helping us, to try to predict or to correlate better all those different layers of information that sometimes, yeah, we're missing. And you might tell me, okay, this is cool, but it's actually very expensive. And yes, I mean, we really like, as put in my intro, we want to really create a social positive impact and really try to democratize all those tools to everybody. So this is also becoming now more accessible, all those automation, all those tools, OpenTrons, we're making some of ourselves. So that's really also making, we want the people in Brazil, for example, who have all those feedstocks, or Mexico, to be able to make their cells, to valorize them themselves. And so that's what we're really aiming in part of my lab. And I think it's a big challenge from industrial biotechnology, but that's a whole different talk. So let's go back then. An example of what we have been doing is we've been working with this anti-cancer drug, Taxol, one of the most widely used drugs. It's a whole pathway, and don't go so deep into it, don't check in the specifics. I just wanted to show all the different drug produced in the plant, we're showing it there, and we want to produce it in E. coli, in Cerevisiae. In this case, we're talking about Cerevisiae. And as you can see, there are so many variables that we can change when we're building a micro cell factory, different genes, different promoters, different terminators, copy number, location, so many possibilities. And apart from that, after you do one construct, so many different variables of how you can test it, how are you gonna grow it, which media and which conditions. So testing it one factor at a time, it's a no, it's impossible. But sadly, if you go to the majority of labs, okay, they don't do one factor at a time, but they may do 3, 4 maximum at a time, and it will never manage to really then optimize those cells. So here I'm showing the one factor at a time. Traditional way won't work. It's been before shown, I mean, that's not a new thing, design of experiments complemented that well with the high throughput. It's the way to go. And I just want to emphasize that's a new way of making experiments in those situations. So we're starting a little bit some small shake flask, and then we go to the microplate, and then yeah, we're scaling up. So here I'm showing some of the data that we're getting. I don't want you to go very deep into what's happening here. What I want to show you is how quickly then, yes, we could get we're getting some of the data of DOE, pH, oxygen, the profile of the metabolites under all those industrial scale conditions. So we could try to detect bottlenecks, enzymes that are not working, and get the next hypothesis. But at least knowing that our cells, those bottlenecks, we're identifying those industrial scale bottlenecks early on into the cycle. And what the ideal thing is, because we want to be able to model. We want to be able to model what is happening with those cells. And here we can see we're managing quite well, at least of biomass, consumption of the sugar, could be ethanol, oxygen still a little bit, but our product formation, we could model it quite nicely using the simple, still the deterministic models, Monod, Michaelis-Menten, but there are always going to be some noise, things that we cannot yet model properly, and that's where machine learning or hybrid modeling has been really useful for us to try to close that gap of the things that we can still not really understand, but we need to be able to predict it. And so not going really deep into that, but that's some of those big developments. Is sometimes it's not about artificial intelligence all and forgetting everything else. When you're combining everything, sometimes you manage to get one of the best results. And the idea is, yeah, we want to do this in silico optimization. For example, here our models, the dots are the experimentals, but we're predicting now with our models that if we tweak a few a few options, a few design conditions, we could get 5 times more of our product. So we're validating now our models to see how, yeah, and we believe that's extremely useful. But still, there are things that we don't understand in industrial scale conditions, or when we're using real feedstocks. For example, unexplained behavior, we're getting a pseudo-hyphy behavior. Our cells start becoming like a hyphy and super viscous. And at least we're detecting that early on, and there's still lots to learn. Why is that happening? We have many hypotheses, but we need to find out that why. And there's all those unexplained sometimes behaviors in industrial scale conditions that we really try to identify and target and solve it. You know, that we've been working on that. And so to finalize that, I just want also to say that I want to make important that sometimes we just focus on the upstream in the industrial biotechnology, and we don't really integrate with downstream. And I think that's a big mistake, and we need to always try to do that. In our case, for example, we were trying to use solvents to extract our products, and that was causing a big problem when we were scaling up. It was toxic to the cells, it was impossible to purify, it was not environmentally friendly. So since the beginning, we're always trying to do that integrated bioprocessing. For example, we decided to use beads to extract our product. It worked fantastically, but early on, when we did in our microscale tools that were mimicking impeller, how you know, the stress would happen and shear stress, we find out that those beads they would break into small little needles that would totally kill our cells if we were going to put more than 20% of those beads. So that was another surprise that we didn't want to learn that until we scale up, until really larger scale. So we detected that, and now, okay, we are then the testing systems, or we're normalizing the beads, we're using some continuous flow systems. So it's important to also then go in that integrated bioprocess to avoid those. And to finalize, and where is the bigger trend is in the automation, artificial intelligence is, as I said, we're doing this loop, in this automated loop, but what if we could let the robot analyze in real time the data and take the next decisions, create new hypotheses, new iterations? We really believe then that will really unlock the power to really create those microbial cell factories. So that's something that we're working on, and we believe that could really then revolutionize what we're doing. So concluding, yeah, so SynBio tools are already mature enough to engineer biology, but we need to really test them at industrial scale conditions to really make real products. Automations, AI is going to revolutionize the way we're doing that, and also low cost will really democratize access to all those tools. And then, okay, I just wanted to acknowledge all my PhD students and my visiting MSc students and all my collaborators for this. So thank you very much. I think I just 1 minute more, and yeah, so that's it for my presentation.
Thank you very much, Leo. That's really exciting, so forward-looking. So we'll have questions at the end, but it's my pleasure now to move on to introduce the second speaker, Katalin Kovacs, who's from the Synthetic Biology Research Centre at Nottingham.
Thank you very much for the organisers for inviting me. My name is Katalin Kovacs, and as I was introduced, I'm working at the Synthetic Biology Research Centre in Nottingham. And today I'm going to talk about one of my favourite organisms, Cuprividus necator, which is gram-negative proteobacteria. It's been found in soil and fresh water. And we are very much interested in this organism because it's a chemolithotrophic bacteria. It can grow on organic substrates, but more interestingly for us, it can grow on carbon dioxide and hydrogen as sources of energy and carbon. Because it's an anaerobe, it can reach very high cell densities, up to 200 grams cell dry weight per liter. And it's best studied because its ability to accumulate polyhydroxyalkanoates, namely polyhydroxybutyrate. It is an industry relevant organism, and it has been used to produce various chemicals such as polymers, ethanol, ketone, isopropanol, fatty acid, and most recently sugars from CO2, which I find very exciting. So as mentioned before, Cinecartor produces polyhydroxybutyrate as a carbon storage compound. It can accumulate up to 80% of its cell dry mass under nutrient limiting conditions. It uses its 5-CAB operon. Other organisms use various different operons to produce polyhydroxybutyrate. Unfortunately, the key thermophysical and mechanical properties of this biopolymer are not ideal. It's a bit crystalline, brittle, less elastic, difficult to process, so it also has to be blended with other chemicals. But it has been shown in the literature that altering monomer composition can improve all these properties. So within the Synthetic Biology Research Center, we're very much interested in, as Anna mentioned in the introduction, in developing technologies that would fit the circular economy in terms of reducing greenhouse gas emission and pollution. So ideally, what we would like to develop is a chassis organism which uses cheap and plentiful resources, for example, flue gases or gasified molecular solid waste, and renewable green hydrogen as food instead of other carbon sources that might compete with the food chain. So we envisage Cinecartor to be used as a main organism to produce various different compounds, monomers, and they can be further polymerized either with PHB or on its own. I'm going to give you just one example, given the short time, the 3-hydroxypropionate work that we've been doing for a while. We also established fermentation process to produce polyhydroxybutyrate, which again can be depolymerized and repolymerized with various other monomers. We also developed, and I'm going to give you the 3HP example again, in this process, production of copolymers like 3-hydroxypropionic acid and 3-hydroxybutyric acid polymers. And within a European project, we also developed fermentation processes using volatile fatty acids, which again can improve the biopolymer properties. In this case, I've just mentioned the polyhydroxyvalerate, which has been shown to have much more superior qualities to the PHB. And all these different polymers then can be used by the consumers. They're also all biodegradable. They can be sorted, recycled, but even if they're not recycled and they end up in the landfill, they can be gasified again and they re-enter the system. So this is how we envisage this wonderful organism in Nottingham. So as I mentioned before, I want to give you an example about how we envisage to produce 3-hydroxypropionic acid, one of the target chemicals, which are currently only produced from petrochemicals. And obviously there's a huge need for this, not only for polymer production, but for other uses. And the first thing when we started working with this organism, we realized that it's not only growing on CO2 and hydrogen, but on pretty much everything else. So producing anything with using this organism is quite difficult, challenging, because it tends to use all the compounds up as carbon source. So we spent several years not finding the genes and the pathways that we had to knock out in order to solve the metabolism of 3-hydroxypropionic acid, which we managed to do. And later on, as you heard from Leo, we assembled lots of different we use a combinatorial approach for each of the enzyme steps. So I'm showing here a 5 enzyme step pathway for 3-hydroxypropionic acid synthesis that we did a step-by-step approach, doing for each of the steps, optimizing up to having 17 to up to 20 different combinations, testing them by feeding the intermediates and then selecting the best combinations to determine where the bottleneck is if we put the whole pathway together, which is often the case. And we noticed that aspartate, for example, in this particular pathway it was a serious bottleneck. So we had to screen for a lot more aspartate decarboxylases. And then eventually we built several different combinations. And as Leo mentioned, often not the building these pathways, the challenging part, but testing all of them and growing them under relevant conditions and so on. But we managed to exemplify that this pathway indeed that proceeds via beta-alanine can be achieved in Cinecartor. albeit we still have some bottlenecks that we need to resolve. And yes, some of these bottlenecks can be eliminated by doing different promoter strengths, different RBSs, different enzymes. But most of the time, like in the case of the C. necator, the bottleneck seems to be, for example, in this case, the intermediates being used up as additional carbon sources by the organism. So we now know that the aspartate is going elsewhere, not just in our pathway. And we identified some major genes using RNA-seq. So we can hopefully further improve our production. In parallel, we also started to look into whether we can incorporate this monomer directly into the polymer, making use of this wonderful ability of the organism to do the polymerization within the cell. So since the most efficient step was from beta-alanine, we tested these property of the bacteria by feeding beta-alanine. And we noticed that most of the 3-HB is secreted in the supernatant, but a more percentage, up to 1.5%, is incorporated. But that's very little. It has been shown that we need at least 10% to make any difference in the thermal and mechanical properties. But ideally, we would like to create a range. So we thought about two potential solutions. First, to test several different CoA ligases that might be more specific for a 3-carbon molecule, and also to overexpress and test other polymerases that might be more specific for this particular reaction. And again, we set out, we built several different combinatorial approaches using 5 different CoA ligases and the 2 polymerases. And in the first iteration, as you probably heard by now, in synthetic biology, you have to do a lot of build test cycles. We got up to 10%, which was very good. We also seen that if we change the operon structure, you get slightly different results, but we were very happy. So we went on to then put a constitutive promoter in front of the best CoA ligase, and that seemed to have improved a little bit. We also noticed that the secreted 3-HB is getting less and less. In the next iteration, we put also a polymerase downstream the CoA ligase to see whether this further improves. And indeed, the results were much better than we expected. We got up to 88% 3-HB incorporation into a polymer, which was really brilliant. Unfortunately, the strain which produced this high level was growing very poorly and produced very little 3-HB. But the strain, which I call here 17, is produced up to 77%, which was great. And this is just again to show that the growth difference between the 2 strains and how this severely affects the pH accumulation and productivity. So ideally, you want a strain that grows well and produces the polymer, not only just has a high percentage. Then we tested a different polymerase from Comobacterium, which has been shown that it works better with a shorter chain carbon molecule, and indeed this further improved. So we are up to 80% now, and it further decreased the amount of 3-HB that was seen in the media. So we managed to activate the 3-HB and incorporate it into a polymer, and we got good yields in this case. So we were very happy. So we progressed from 1% 3-HB in the first instance to about 80 more percent. So you might not think that this is a great achievement, but previously the highest amount of 3-HB incorporated in a polymer was 7% when the organism was fed with 30 grams of 3-hydroxypropionic acid. So just to put it a little bit in a context, this is really great. We also wanted to check whether we can produce a range by feeding different level of beta-alanines, and indeed that's the case. And we were interested in this because we also have strains that produce beta-alanine, which we can control with an inducible promoter. So that's great. So we can then further test the construct in the background strain, which again had to be engineered to, so it can't use beta-alanine as carbon and nitrogen and energy source. But we also were interested to see whether if we do any genome engineering, whether that would further improve our polymer. And indeed, we deleted the genes in the main polymer synthesis operon, as I mentioned before, FASEAB, whether they are homologs in the organism. This operon is the operon mainly responsible for the production of the polymer. So we created these strains and we tested the best, the 21, the best combination, and indeed this time we managed to produce up to 91% more percent of 3HP with our biopolymer. And interestingly, this polymer also showed reduced polymer turnover. So for those who are not familiar, obviously these organisms accumulate these storage compounds. So when nutrient limitation kicks in, they can use it as carbon source. But obviously this new polymer, they're not, so their depolymerases are not able to access it as well as the polyhydroxybutyrate. So that's good as well for an industrial purposes. So just in conclusion, and just to give you an idea what we're planning to do in the future, we've shown that we can implement a pathway for 3HP production from central metabolism. We generated polyhydroxybutyrate, polyhydroxypropionate copolymers with a content of 0% to 91%, which is great. We would like to do this now from CO2, as I mentioned before, from strains that produce beta-alanine and so on. And finally, we have done already some of this characterization, the thermal, physical, and mechanical properties. So we are just moving on to getting these new novel polymers characterized, and because the mechanical properties are not yet known for these polymers because they did not exist previously. So we're very excited about finding out, but we already know that they are very interesting because they are transparent, they are elastic, and they look quite interesting. So with that, I would like to thank you for the supervisors, for Alejandro, Callum, and Christian, who's done a lot of the strain engineering work, fermentation scientists who help us a lot with setting up all this, the genome scale modelers and the modelers who always advise us of which genes to knock out and how to direct the carbon flux and the energy needs and the hydrogen needs for our products, and obviously the analytics teams who do all our GCMS and HPLC analysis, and everybody in the lab. I would also like to thank the funding bodies. Thank you.
Thank you very much, Catalin. As I said before, we'll have questions at the end, but thank you for a lovely presentation, and we'll move on to our third speaker now, Mark Dunstan, who's from the Synthetic Biology Research Centre in Manchester.
Okay, so well, first, thank you very much for the opportunity to speak today and tell you a little bit about what SynBioChem have been doing over the last 18 months or so. So SynBioChem has been running for about 5 years. The centre's ambition was really to innovate, develop and integrate a suite of technologies for the synthesis of fine and specialty chemicals. And we modelled this on a biofoundry concept using the design, build, test, learn cycle that everybody's quite familiar with. So the pipeline we design must be agnostic to the target compound as well as the host organism, but also it must function with no assumed pathway knowledge. And we use a lot of automations that's to improve performance and again to reduce the human error. And ultimately this was to provide software, platforms and expertise for the wider synthetic biology community. And we applied these to 3 core projects, flavonoids, terpenoids and alkaloids, and I'll come back to the flavonoids a little bit later. So in terms of the pipeline, I don't want to go into too many details. We've presented the pipeline sort of many times before, but for people that are not familiar or are interested, we do have a paper a couple of years ago. If you want, please go and have a look at that. So what was asked, one question we were asked is how many pathways or compounds can we do at once? And even though this is quite a simple question, obviously we put quite a few compounds through and done a number of pathways. We've never really multiplexed. It was quite a difficult question for us to answer. So anyway, a few days after this question was asked, we had a meeting we had no idea what the meeting was about, but this turned out to be day 0 in a pressure test, and this was designed to answer this very question. So the brief was, could we use our current pipeline and use it in alternative chemical spaces that we have no prior experience of? For this time we chose material monomers, we'd never worked on these. And can we tackle a large number of compounds? at once and achieve quantifiable target products in a short time frame. And we chose around 85 days, and this took us up to the Christmas period. So around less than 3 months we had total. And I just want to stress this time, the time frame also includes not only to select the targets themselves, but also includes all the design genetic parts, as well as gene synthesis as well. We know it can be 6 to 8 weeks. And hopefully we'd use these metrics to look at the bottlenecks, the limitations in our pipeline, and improve it, maybe make a pipeline version 3.0. So after this meeting, that very same day, the team went away and we looked at the top 30 bio-based platform chemicals and those in the shikimate factory, and we tried to identify industrial relevant monomers or novel compounds with similar functionality that we could use. We sort of eliminated endogenous E. coli metabolites such as lactic acid at this stage. So we came up with 27 material monomers. These were from 7 diverse classes. So we got vinyl benzene, so for example for vinyl phenol, which is used in LCD displays. Mandalates, so polymandalide is degradable polystyrene mimic. Diols, for example, flame retardant polymers, for example. Dienes in rubber, synthesized in rubber, and some like dicarboxylates for like monoclinic acid, which is a precursor to nylon production. And to cut a long story short, the team worked extremely hard over the 3 months, and I think we calculated that we spent 360 personal man hours on each material monomer. As it turned out, the project was really successful. We managed to make 17 material monomers from the 5 classes, and these were produced in vivo over a 30 to 84 day period, and these are highlighted all in green here. And in many of these cases, the titers are actually close, in some cases a little bit better than the reported optimized E. coli fermentation conditions. We also looked at, so in blue we've highlighted E. coli metabolism. We also looked at developing 9 chassis in the end to enhance key E. coli metabolism, for example, phenylalanine shunts and tyrosine shunts. And these proved really useful not only in this pressure test, but also for our core projects, which I'll come back to a little bit later. Of course, there were failures, and I'll just talk through some of those. There was failures, dropouts throughout the pipeline, as it turned out. So, for example, the phthalates, the decarboxylates, the phthalates, we struggled to get enzymes to do these reactions, so they failed at the enzyme selection stage. In some cases, we could build the platform. We could build the platform, the pathways. Unfortunately, in this case, for sulfuric acid, we could build the pathways, but we couldn't get a product. And in some cases, we could find the enzymes, but it wouldn't build for whatever reason, maybe toxicity or biological reasons. And in some cases, unfortunately, for the isobutyrate compounds and the dienes, the problem was that we couldn't get detectable mass spec methods in time. In this short time period, these are quite volatile chemicals and were quite difficult to get methods for in that time frame. But having said that, still a very successful project. And just to highlight this test in numbers, over this 85-day period, we managed to make 135, design and synthesize 135 genes. We put these in almost 200 unique pathways. We also developed an enzyme screening pipeline, and we screened 142 in vitro enzyme. We had 17, like I said, we made 17 compounds over the 5 of the 7 classes. And in that time period, I think we run something like 8,000 mass spec samples over 22 different methods from our different instrumentation. But we didn't want to really stop there. We'd made all these chemicals, and we said, well, can we now scale these? Can we make them into the gram per litre scale? And we used a number of techniques, for example, directed evolution. We looked at pathway optimization. We also looked at process optimization, so for media optimizations and fermentation conditions, and we looked at strain engineering. And not only were we able to make them in gram per litre quantities, so for example, here we've got mandelic acid and hydroxymandelic acid. we made these at 1 gram to 4 to 5 grams per litre. We're also able to make stereoselective strains in which we could make S-mandolate, R-mandolate, and the equivalent S and R for hydroxymandolate. So again, a really successful pilot scheme at scale for some of these compounds. We then looked at what we'd learned from the scale up and said, well, can we apply this to our core compounds, our core projects? So here I'm going to talk a little bit about flavonoids. I'm going to give you a quick, little brief introduction. So flavonoids are plant-based. They've drawn significant attention over the last few years as dietary supplements, and particularly the 2S flavonoids. They've proved to be an important subgroup and have wide-reaching applications in health, from sort of blood pressure to anti-inflammatories to anti-cancers targets as well. And these flavonoids are present in numerous sort of fruits. They're also available. There's lots in you find in red wines. So I'm sure that our flavonoid intake has increased quite a lot during lockdown. So we looked. What our flavonoids we aimed to look at was we looked at naringenin, phenacimrin, eridictyl, and omeriodictyl. And the reason we chose these because they occupy a central position as key branch point intermediates towards a broad range spectrum of natural occurring flavonoids. And you can see here we've got naringenin and phenacimrin. You can see this is just one enzymatic step away, and you can see we get a diverse range of flavonoids that we can access. So these flavonoids are currently extracted from plant-based sources, so grapefruit pulp, for example, for naringenin, or they're chemically synthesized. Now, both of these have their downsides. So, for example, so extracting from plant-based, there's actually quite poor abundancy of flavonoids in the pulp, and also we have inconsistent harvests, which makes the cost of these flavonoids sort of fluctuate quite a lot. And also during chemical synthesis, use very toxic solvents, or they're not extreme chemical reactions, which doesn't yield a particularly green product. And this can be problematic for downstream applications. So you're using it in, for example, for dietary supplements, it'd be quite difficult to get to translate to that. So to produce these flavonoids, well, we'd already produced phenacimrin in year 2 at about 50 mg per litre. So phenacimrin is a 4-gene pathway. It comes from phenylalanine and uses a PAL to go through to phenacimrin. Naringenin is very similar. It uses the same pathway, except it comes from tyrosine and uses a TAL. So what we thought we'd use is the architecture for our previous phenacimrin pathway to build a library for naringenin, and we looked at used our enzyme selection tool, cell enzyme, to select a number of 4CLs and TALs. We put them into a small library, a 15 pathway library, and 4 of our libraries produce 75 to 90 mg per litre of naringenin, which is a really quite nice start point. We then took our 4 best pathways and we tested against 9 common E. coli strains. And although the lineages of these strains are very similar, we do see quite remarkable differences in their ability to produce naringenin. And we had a clear producer like the Danish Hive Alpha produced around 300, which is clearly better than any other chassis we tested. We then took that our best pathway and our best strain, and we looked at process optimization. So what can we do with a media, our carbon source, etc. And we tried several carbon sources, but the best was glycerol in combination with TB, which gives a base strain producing around 350 mg per litre. Now, at this point, we are still kind of really cheating in the terms that we're actually also giving feedstocks here. So we're actually also adding tyrosine and a fatty acid inhibitor called cerulenin. So the next obvious step was, well, can we remove these expensive additives? And if you remember in the stress test that I've just talked about, we actually made a double mutant, which was to sort of increase tyrosine production, if you like. And we also had a 3-gene tyrosine shunt pathway that we'd integrated on the genome, but we also have a standalone plasmid for this as well. So we applied this to our naringenin production, and as you can see here, we've got the double knockout with our plasmid-borne tyrosine, and it more than compensates for our wild type with the addition of tyrosine. So we have addition of tyrosine and our double knockout and tyrosine shunt without tyrosine added. So now we got to the point where we could remove the tyrosine addition. Now we thought, well, could we actually now go and remove the need for cerulenin, which again is a fatty acid inhibitor. So we came up with this really neat multiplex CRISPR-I system where we could target numerous genes in the fatty acid biosynthesis. And if you can see here, we have a multiplex system where we have up to 5 genes. And here we have in the green our wild type strain without cerulenin, and then the yellow and orange where we've added cerulenin in. And you can see that with our multiplex 5 gene CRISPR-I system, our knockdowns, you can see that we can more than compensate again for the addition of cerulenin in our wild type strain. Okay, so we made a base strain for naringenin. We then applied this to pinacemerin. Again, we were able to make a 3-fold increase using the same genes we'd used in the naringenin pathway. So now we have 2 base strains producing 500 and 200 mg per litre. We then turned our attention to eridictol and omieridictol. Again, I mentioned in the pressure test that we'd made fluoric acid pathway that didn't work. We knew what was wrong with that, and we were able to make a successful pathway producing fluoric acid at 200 mg per mil. And using this pathway with a 3-gene version of our flavonoid pathway, we were able to produce both eridictol and omieridictol. So just to summarise, again, over the last 4 to 5, year 5, we managed to multiplex the system. We managed to use a completely new research area for those materials and produce 17 of these in 85 days. We're able to scale up these compounds, and I think this really showcases the ability of biofoundries to provide a quick access, not only to the virus range of material chemicals, but any sort of target molecule. And demonstrates how prototype microbial production strains can really rapidly scale up and can be optimized to achieve that gram scale fermentation. Again, we also applied this to our flavonoid to make 4 key gatekeepers, and these strains provide a really nice springboard for us to produce a range of both natural and non-natural flavonoid production. So just leaves me to thank people who did the work, the directors who make us do the work, and the funding bodies. And thank you very much for your time.
Thank you very much, Mark. That's a really exciting presentation. So we now have an opportunity to put some questions to the speakers. And so I'm going to have a look through the question box here. I'll ask Catalin a question first. This is from Naglis Malice.
Okay.
Who says companies BASF, Cargill and Novozymes have already claimed efficient production of 3-hydroxypropionic acid almost a decade ago. How is C-Necator better for 3-HP production, and what yields can be achieved using C-Necator and carbon dioxide as a carbon source?
That's a very good question, and indeed, not only E. coli, but Cerevisiae, the yeast, and Klebsiella, they've all been used and industrially to produce very high yields of 3-hydroxypropionic acid, but they all use glucose as carbon source, which obviously that is something that we do not want and we want to avoid, because that would compete with the food chain, and as we know, that's food is just as big as an issue as the lack of petrochemicals. So that's the first part of the question. Secondly, the reason we would like to use C-Necator, because we could use carbon dioxide and hydrogen as a carbon source as a carbon and energy source to produce this. So obviously this would help with reducing the greenhouse gas gases as well. As for the yields, it's still quite tricky to tell, because at the moment, if you remember my slides, the yields are not very high, but still only millimolar range compared to grams per litre in E. coli, several grams per litre in E. coli. we have done some calculation using the genome scale models. There are some maximum theoretical yields. They are not bad, if I'm honest with you, but we have to see what we get. We haven't done the whole pathway from CO2 yet. We're waiting for the fermenters to become available.
Okay, thank you. So now we have a question for Leo from Vasily Ramsey from Malaysia, who says, can you please share what type of data you use for in silico optimization of the Taxol biosynthetic pathway? Is it based on flux, metabolites, or gene expression data?
Okay, so we combine as much as we can. I mean, first is based on bioprocess data too, like cell growth and the gas of the bioprocess, and then it's more the omics data, metabolomics data, could be transcriptomics data, could be the proteins concentration data that we are using to do all this modeling. So that's how we try to then that we manage to predict and then try to do this in silico prediction.
Okay, thank you. And I had a question for Mark with your Grand Challenge project. Was DNA synthesis a bottleneck in any of that?
So DNA synthesis certainly is, because it's 6 to 8 weeks roughly. I mean, it is coming down quite a lot, but we did find that one of the real bottlenecks that we hadn't realized was that actually it was processing the parts. So we realized that making the genetic parts, the PCR, the cleanup, it wasn't really particularly that automated. And when you're looking at 200 to 300 genes, it really became the bottleneck. And because you're building so many pathways, if you need all them parts. If one's missing, then you can't build that particular pathway. So it was not only was the gene synthesis, the part preparation. This next probably bottleneck that we observed was the analytics, really, to sit there and analyze the data that was coming off. I think it took longer to write the paper than it did to do the experiments.
Yeah, so I'm not asking you to advertise any DNA synthesis companies, but you managed to get that done quickly, because clearly if that's a hold up for a Grand Challenge, it's a major problem. Yeah,
I mean, we do use Twist. Well, not to advertise, but yeah, they're cheap and they're probably quickest that we can get. And we've got a system set up where they can put it straight. So we build our BioBlock and they put it straight into sort of a plasmid that we can access straight away. So it's quite convenient for us.
Yeah. Okay, I have another question for Catalin from Simona Della Val. Are there any standardized DNA assembly methods for use in C. Necator to enable rapid generation and testing of different constructs?
Any standard assembly method you could use. We use HiFi assembly. Some people in our group use user assembly. Our preferred option is HiFi or Gibson. I find that's very easy.
Right. Okay, I'm just looking through the questions here, just give me a second. Now, there was an interesting one about AI. I can find it. Basically about whether there was, I can't find the question at the minute, but whether you've considered the idea that there could be an invented step in using AI in your approaches, and what that might mean for protection, IP, commercialization.
Okay, well, I don't know.
Have you considered future commercial implications of saying that your hypotheses are invented by AI? That's from Sarah Holland.
Okay, that's a very interesting question. I mean, yeah, there's so many things when you bring in artificial intelligence that is difficult difficult to answer, and that's why we have incorporation of social science. There is ethics, and that questions you're right, So actually, good point. That's a question that we have to talk to our business managers, and this is a new area. So what we do is we discuss with the business manager, and they tell us, how to protect it better But that's totally a good point, like I'm sure they're gonna be puzzled too if we tell them, that the discovery was not done by us, but the robot. You can technically, I would say that we created the robot, we created the system, so it would be still our discovery. But there is this interesting ethical, or no, I wouldn't say ethical, but this interesting question, that.
Yeah, there might be a robot on a Bahama cruise ship somewhere, if that's what robots like to do, off the proceeds of inventiveness. Well, unfortunately, there are lots more questions, but we've run out of time. And so I now need to wrap up. But I'd like to thank the speakers very much for their presentations. I think it's been a real shake-up in terms of the way we think about doing things. And we can continue the conversation online. So you can follow at BioChemSoc and at PP Publishing on Twitter and tag them in your comments about the talks you've heard today. Last week we heard from other researchers working in the field of synthetic biology. And this time of year we usually meet at Synthetic Biology UK, but for obvious reasons we're not able to, which is a real shame. But it's great to have so many members of our community come online to enjoy this content, and we hope we'll be able to see you next year. So you can register for SB UK in November 2021 on the Biochemical Society website, and the URL is on your screen now. Next week in the Biochemistry Focus webinar series, we turn our attention to the sociology of the anti-vaccine movement, with a presentation from Professor James Cherry of UCLA and chaired by Professor Rosalind Smythe, Director of the UCL Great Ormond Street Institute of Child Health. And you can register for this, watch past webinar recordings and find out more about the series at biochemistry.org/webinars. On that same webpage, you can also propose your own topic to be hosted in 2021. So I do encourage you to think about that and think about which areas you think this series should cover. Lastly, I just want to highlight that in these challenging times, it's more important than ever to stay connected. It's an extraordinary time for us all, but it's also an exciting time to join the Biochemical Society's community of researchers and specialists to stay connected and take advantage of key benefits, including discounted registration fees for society courses and meetings, exclusive access to a range of grants and bursaries, personal online access to 2 of their journals, and more. So have a look at their website to find out about membership at biochemistry.org. So other than that, I'd just like to say thank you very much to everybody, to the speakers and to the people who've registered and listened to the presentations and ask questions. It's a shame we can't do it in person, but thank you very much. It's been a really good event. So bye everybody.
Thank you very much. Thank you. Bye-bye.
Bye.